Haifa, Israel. Daniel Aberdam is our host speaker today. He is an expert in skin and corneal pathophysiology. His research focuses on understanding the molecular mechanisms of eye and skin diseases and the search for potential therapeutic tools. More recently, his group has developed an in vitro model of Aniridia-associated keratopathy. Daniel currently works as a director of research in INSERM, Paris, France. Well, just a little housekeeping before we get started. If you have any questions during the presentation, please send them through our social media channels. I'll bring them up during the time for questions at the end. With no further ado, we will turn the time over to Daniel. Welcome.
Okay, thank you very much, Juliana. So it's a real pleasure to introduce Professor Ruby Shalom Feuerstein. We'll call you Ruby. He's now an established professor at the famous Israel Institute of Technology, the Technion, which is located in Haifa in Israel. I was lucky in the past to welcome Ruby as a postdoc, a young postdoc fellow after his PhD thesis at Tel Aviv University. I welcomed him in Paris, in Nice first, and then in Paris. And actually, we became also good friends. His work focused mainly on basic research. He is trying to understand how epithelial, like skin, cornea, as example, can be formed or new by specific stem cells. And as a consequence, what are the reasons for disease, for pathologies? And for that purpose, is using stem cells technology, but also animal model, and also algorithm and mathematical models sometimes. So the last years, his team has really improved our understanding and how cornea can maintain transparency, how the cornea stem cells are able to renew the epithelium along our life, and what contain the niche that maintain the existence of the stem cells. And all this knowledge on normal situation will help us, of course, to better understand the disease, for instance, Aniridia, and maybe to find tools, molecules that could treat patients and allow them to rescue their vision or to help not losing. So I'm really proud to invite Ruby now to present his talk entitled Eyes Open on Stem Cells Dynamic, Signature, and Niche. Thank you, Ruby.
Hi, thank you. Thank you so much, Juliana. Thank you so much, Danielle, and everybody that is attending to this session. Just by the way, for curiosity, Juliana, I don't see any of the audience, but I don't know if I'm supposed to see. So just to know if I should see somebody, or is it normal?
You are not supposed to see anybody. We are being webcasted through YouTube channel, so you won't see anyone in the
like a researcher. You feel alone and you're doing your research and also the presentation. Do you see my screen, by the way?
Yes, I can see it.
So, okay, so thank you. I'm so happy for this opportunity. I'm really honored for this welcoming and to be presenting in this webinar that is super important, I think, and the contact with patients is also super important. I must say that, so my lab is, as you said, is focused on stem cells. So in general, we're addressing questions, key questions and fundamental questions about stem cell biology in our body. And as a model, we're using the cornea, which is our favorite model, or the skin, which is also a model that we use quite a lot. We typically work on transgenic animals, and we harness the transparency of the cornea in order to see the cells and to look at their life of the cells, and especially the stem cells. So if you can see my cursor, please confirm, Juliana, if you can see my cursor now.
Yes. I can see it.
Right. So you can see that the cornea, this dome-shaped tissue, is very interesting for us from the aspect of stem cell biology. And this is why and we're trying to harness the advantages of the cornea for research. So what are stem cells actually? From the moment that the egg is fertilized and the entire body is slowly formed, and you have the beautiful structures are formed, all the tissues and organs are formed, but still, nevertheless, even though morphogenesis has been completed in the adulthood, we still need a lot of cell replacement. So every day, our cells, our body is producing millions and maybe more of cells every day. And for that, we need stem cells. And the definition of stem cells and their hallmarks is their ability to proliferate and to self-renew. We call it self-renewal, and we mean by that that stem cells can renew their pool of stem cells. They can generate copies of themselves, or otherwise, they can also into specific cells that have tissue-specific function. So in our adult body, we have different organs and tissues, and in every tissue, it is believed, probably virtually in all tissues, we have, or nearly on all tissues, we have stem cells that are specific to this tissue, and they are limited in the way that they can only produce cells for that specific tissue. For instance, gut stem cells can produce gut cells, like enterocytes. These are the cells that are able to absorb the food and to process this metabolism, bring it into the liver. So this is a specific feature of gut stem cells. They can produce all the gut cells. And similarly to the cornea, limbal epithelial stem cells are able to generate the limbal epithelial and corneal lineage. And actually, the traditional stem cell dogma is long-standing. It's been 60 years since it was proposed by Thiel and McCulloch, where they, in the early days, proposed that stem cells are rare cells within the tissues. They are long-lived, meaning that they would stay for long-term, and they proliferate very infrequently, and this saves them from accumulating mutations over our lifetime. But once they proliferate, they produce progenitor cells. These are short-lived cells that are usually sharing the same niche with the stem cells, the same microenvironment. And these short-lived cells are fast-cycling, but they have only the ability to proliferate few times before they terminally differentiate and lose their ability to proliferate, become post-mitotic differentiated cells. And these are also important cells. These are the cells that are doing the function usually in the tissue. They're expressing specific sets of genes that is important for the tissue function. However, this hypothesis was, although it was supported by many other tissues, it started in the bone marrow in the 60s, later on the skin, stem cells were discovered and studied, and many other tissues supported this dogma that stem cells are rare, and everybody, including us, was trying to find these markers for these rare stem cells in order to harness their potential for therapeutics, because of course, for every tissue, we would like to isolate these stem cells, purify, be able to grow them, and be able to modulate them if they are mutated for the benefit of patients and for the better understanding of how tissue functions. However, despite this many effort that has been done, it was very difficult to identify markers that distinguish the two cell types that can proliferate, okay, the stem cells from the progenitor cells. And most of the markers that was proposed labeled too many cells. Okay, they labeled probably, if you like, both stem cells and progenitor cells. And actually in other tissues, in few tissues, there's been reports that, or studies that reported the absence of infrequently dividing, or what we call slow cycling cells. And also there was lack of evidence in many cases, or not convincing evidence, for small niches that would support these isolated islands of stem cells within tissues. So stem cells in a way remain still enigmatic still today. There's a lot of controversy, but despite all that, we're still able to use stem cells, even though we cannot purify or identify or predict which cell within the population is a stem cell. We can still harness stem cell for stem cell-based therapies by bone marrow transplantation. Even if we don't know which cell exactly is a stem cell, we can transplant bone marrow cells or skin cells in order to graft it to patients with skin burns. And also corneal or patients that suffer from a chemical burn or from mutation may suffer from a disease that is called stem cell deficiency. And this terminology implies that, or proposes that, the loss of stem cells was the reason for the opacification of the cornea and the neovascularization of the cornea. And actually the therapy for these patients is based on the grafting of limbal tissue or limbal cells that are expanded ex vivo and then grafted back to the patient. So we can still have hope to progress, but we understand that we have some obvious challenges that are limiting us and limiting the application of stem cells for the benefit of the patients. We focus on the cornea because the cornea is a fantastic model of stem cells. So the cornea, I guess most of the audience know it well, is a dome-shaped tissue that protects our eye against external insults, including microorganisms, toxicant, and so on and so forth. And it serves as a barrier, but also allows the tissue to focus the light onto the retina that is behind. And actually, the cornea is doing its property of a barrier to large extent thanks to the epithelium that is the outermost tissue. This is the structure of the tissue on the right in this scheme. So you can see from the outermost tissue, it's a stratified epithelium. It's a wall of cells that is protecting us. And these cells are supported by a structural tissue that is called stroma. And this tissue contains mostly fibroblast-like cells called keratocytes. And if you look at the zones of the cornea, you can divide it into mainly two zones, if you like, especially in the perspective of stem cells, because it's been shown by many accumulating evidence that the limbus, the boundary, this ring-shaped zone between the white part, the sclera that is covered by conjunctiva, and the cornea, this ring-shaped narrow zone hosts the stem cells. So according to the dogma that I just showed you in the previous slide, these stem cells are rare. According to the dogma, they proliferate and they can generate progenitor cells that are short-lived. And these cells are slowly moving to replace and renew the corneal epithelium. This is very important. They are moving this direction, okay, or here in this scheme, this direction, while they continuously start to more and more differentiate, and then they delaminate. They leave this basal layer. Basal layer in the limbus and the cornea is the place where cells can proliferate. When they leave this basal layer, they become postmitotic and differentiated. They cannot proliferate. Eventually, they will be shaded off the tissue and die, and they need to be replaced. But despite this very strong dogma, there has been a publication in 2007, then in 2008, very nice data, I must say, that was also very controversial, showing that grafting of a limbus that was labeled, here it's shown in blue, in mouse, did not give rise to radial movement of the limbal graft. One would expect, according to the dogma, that if limbal cells and stem cells are located in the limbus, and if these cells are the ones that are important for the regeneration of the corneal epithelium, one would accept that after long term, after this grafting, these cells would migrate to the center of the cornea. Moreover, another evidence was quite striking was that the ablation, depletion of the entire limbal epithelium of the mouse did not cause the so-called limbal stem cell deficiency. This opacification of the cornea was not evident, even though 140 days and even more was shown in that paper. There was no evidence for this opacification. And actually, they proposed that stem cells do not localize specifically to the limbus, but actually that they are localized to the entire, they are distributed over the entire corneal epithelium. So our goal when I started the lab was to establish a vital stem cell tracing in order to reveal stem cell location, and then to use this model in order to study the dynamics of stem cells. And in order to do so, and also to distinguish between the two hypotheses, whether the stem cells are restricted to the limbus or otherwise, if the stem cells are distributed all over the tissue, we decided, with the work of Aya Amitai and Anna Altschuler in my lab, we decided to take an approach without grafting. If you want to study stem cells in normal conditions, it's not recommended to perform injury and grafting, which does not reflect the normal condition. So actually what we did, we used an approach of lineage tracing where we label specific, or actually not specifically, but randomly, we label the proliferative components of both limbus and corneal epithelium, okay, with stochastic expression and a random of 1 out of 4 fluorescent gene within every cell. So you don't need really to understand the genetics if you don't want. The only thing that you need to understand is that before we inject these double transgenic animals, before we inject them with a chemical tamoxifen, the cornea would be dark and it would not fluoresce. But once we inject the tamoxifen a few times after 3 days of injection, we can see that many basal cells become positive, okay? And they become stochastically positive to different colors. And this color is stable in the DNA. So the cells, if it proliferates, then it will give rise to many cells with the same color. Okay, so the hypothesis was that if the stem cell location is in the limbus, we're going to see a kind of a limbal stripe kind of cornea if we trace them after long term. Because you expect these blue stem cells, for instance, to produce cells that would migrate and generate this large clone of many cells and stream cells to the cornea. However, if the stem cells are located all over the cornea, we expect to see a raisin cake, okay, with patches of clones. So as you see, what we saw was that the limbus is the main location of stem cells, or perhaps the only, that's what we believe, it's the only location of stem cells of the corneal epithelium. But it raised the question of how come the entire depletion of the limbal epithelium in that paper that I just showed you in the previous slide, how come it did not give rise to any pathology? So this was quite striking. And in order to address this, we sought for another reporter. So we were looking and we did some search of different transgenes, and eventually we were lucky enough to identify another transgenic reporter. And this is not an irreversible like the other confetti one. This one is a reversible reporter. So you can see we were quite surprised and happy to see a very nice discrete expression of green fluorescent protein in the limbus. And actually, when the cells are leaving the limbus and regenerating or renewing the corneal epithelium, they become negative. Okay, so we thought that probably these green cells are stem cells, or at least some of them, or many of them are stem cells. And indeed, when we did a triple transgenic animal that contains both confetti, double transgenes, and the K15 GFP stem cell reporter, we could see that these green cells are the ones that are streaming the cells and renewing the corneal epithelium, strongly suggesting that many of these green cells are stem cells. Okay, but then we could do the big experiment, the one that we were most interested in. This was a project of Wasim and Aya, where we could now more accurately deplete the entire limbal epithelium. So here you can see before depletion, and this is after the depletion of the limbal epithelium alone, without affecting the stroma. This is histology showing that the stem cells has been removed, the epithelium has been removed, but not the niche, not the stroma underneath. And then we could follow the tissue, and hopefully we wanted to see whether we're going to see the formation of what is called stem cell deficiency, disossification and neovascularization of the cornea, loss of vision, or are we going to see that the tissue is not affected like it has been reported in Nature 2008? And we were quite struck to see that the cornea remains clear and transparent and looking normal for many, many months, for years actually, until the mouse dies. And also we could see that there is a recovery of this K15 GFP signal, and we were quite puzzled. Where are these cells coming from, these K15 GFP positive cells? Where are they coming from? And we did a couple of experiments that I don't have time to show you. For instance, one of these experiments was to remove the entire limbus plus the entire corneal epithelium, both epithelial tissues. And in that case, we have never seen any recovery. And this implied that probably these cells are not from the conjunctiva coming, because the conjunctiva was remained intact. And they were probably coming from the corneal epithelium because this is what we just removed and there was no recovery. So in order to address that, we did another experiment using the triple transgenic mice. And please note carefully about these slides. So what you can see here is a living animal before surgery and after surgery, and also 1 day or 3 days or more after surgery. And you can see here that we did triple transgenic animals. So we have the stem cell reporter, K15GFP. Before the surgery, the cells are there, and they are streaming a stripe of cells. We are looking at one stripe, only one color and one stripe, and this is very helpful to follow over time after many days. You're going to see that we follow them for very long terms. And this is what we saw. So just after depletion, we see that the mouse under anesthesia, it doesn't have any stem cells whatsoever in the limbus epithelium. However, 1 day post this removal, you see that these cells here, they migrate and they cover the denuded limbal epithelium, also the conjunctival that was a bit denuded as well. And 3 days after this movement of the cells, the stripe was a bit thicker. You can imagine that there are many stripes going in the same direction, healing the limbus, but nothing happens, okay? The cornea is transparent. There is no K15GFP. It is not before day 7 or day 10 where all the limbus becomes strongly, in 2-3 days, bursting to more than the normal expression, even twice more. Okay, and then you see K15GFP is very striking and it's very stable, going back to normal over time. By 30 days, it becomes a bit more thin and quite normal to what we're used to see. And this strongly suggests that it is corneal committed cells that are able to heal the limbal epithelium. Even though these cells are differentiated, they can still sense something in the niche and then become K15GFP positive. We did some other staining showing that these are most likely stem cells because these cells express the markers that are the hallmarks of limbal stem cells. So what is the role of the niche in this context? So we could see that if you remove the entire limbal epithelium and also damage the niche by a very localized damage to the stroma, in that case, there has never been any recovery of K15GFP, and there was stem cell deficiency, this opacification and blindness of the mice. It happened all the time in all the mice. We did some dozens of mice, and it always happened. So you must have an intact limbus niche in order to have this dedifferentiation process, so that when the committed cells are coming back to the niche, they must sense something that is good for them and directs them to become limbal stem cells and prevent this phenotype that is seen in patients. The last part of my talk, I will talk about stem cell population that is also related to this K15GFP probe that we used. So as you can see, we were quite puzzled by the pattern of expression. It was a punctuated expression in the limbus. And not only that, it was a bit annoying to see that the cells are not at the edge of the limbus. Okay, there are some cells that are negative to K15GFP, and still this is considered the limbus. So we were puzzled, and we thought that it suggests that there is some heterogeneity in the limbus. And we did a single-cell RNA-seq, like many other groups have recently done in many tissues, and also in the cornea, actually. And we took 10 corneas of mouse, of course. We took the limbal epithelium and some margins of conjunctiva and cornea, and we isolated the epithelial cells, and we subject them to sequencing, and we could have very nice signature of 2,500 genes per cell, more or less, 5,000 cells, more or less, were captured. And the algorithms could unbiasedly cluster them into groups of cells with similar gene expression. And actually, we had in this experiment very nice data showing that there are two limbal basal cell populations that were mostly interesting. This refers, we thought, maybe to these K15GFP positive or negative cells. And also, we could see that there are 4 corneal epithelial cell types and some other cell types that could be also interesting for future research. And but this was only an in silico computerized analysis based on markers that are known for the limbus, for the corneal epithelium, for the conjunctiva, and so on and so forth. And in order to validate where are these clusters of cells that have similar identity, where are they located? What are these two limbal basal layer population? Where are they located in the limbus exactly? And we were quite surprised to see that the outer limbus, we coined it the outer limbus and inner limbus. So there were two specific zones. We did tissue sections and also hole mount. And as you can see, using all markers that you can imagine, we did a lot of staining. One could see that there is a zone that contains outer limbal epithelial basal cells. This is a hole mount looking from the front view. And there is another zone we coined this zone inner limbus that hosts other cell population expressing, for instance, K15-GFP. The best markers actually were of the outer limbus, GPHA2, CD63, and K15, and EFIT-M3 were excellent. If you want to look for these cells, I would recommend to do hole mount. You can also do tissue sections, but best not to lose. It's because it's few cells, is to do hole mount, so you have the whole tissue, the whole cornea is intact. And the best marker to look at for the inner limbal stem cells was K15-GFP, and we could also use other markers. And then the question was, what is the difference between these two cell populations in the limbus? These are two new sub-compartments of the limbus that have never been shown, so we were puzzled about the function of these cells. So we did a couple of proliferation assays that I don't have time to show, but you can read in the paper. It's now online since last week in Cell Stem Cell. And you can see that we traced slow cycling cells by pulse chase experiment, and we discovered, to our surprise, that the outer limbus is the one that hosts slow cycling cells and much less the inner limbus. Okay, it was the inner limbus that hosts these red labeled actively dividing cells. And we did a couple of more experiments to estimate the division frequency of these cells. So the outer limbal cells were dividing slowly every 8 days, not very slowly, by the way, but we still, to keep the consistency with the literature, we refer to them as quiescent stem cells, because these are the quiescent stem cells that are typically shown in the literature. So we think that outer limbal cells are quiescent limbal stem cells, while the inner limbal cells are the active limbal stem cells. Now, so what is the role of each population after wounding? So if you wound epithelium, we see a lot of proliferation everywhere, both in the outer limbus and in the inner limbus, suggesting that both cell populations participate in wound healing process. Now, to follow better and to know better the proliferation dynamics, and to extract some data that would shed light into how many of these cells are stem cells, we did lineage tracing again, and as you can see, 7 days after tracing, you can see the outer limbus and inner limbus. Here is the cornea, and you can see that we traced clones that are very small because it's only 7 days after the induction of the coloring of the confetti. But already after 1 month, you can see that the clones are becoming bigger in the outer limbus, but they are becoming much, much bigger in the inner limbus and in the corneal periphery, suggesting, in line with the previous slide, showing that the inner limbus is proliferating much faster than the outer limbus. And this was even more striking 2 months post-induction. You can see very small clones still in the outer limbus, while the inner limbus, you can already see the pattern of stripes that are streaming cells of the same color, suggesting that it all started from a single cell to the central cornea. And after 4 months, we could see large stripes. Most of the stripes, to our surprise, were coming from the inner limbus, while only few stripes, if at all, were coming from the outer limbus, 3%, and they were coming from the edge of the outer limbus, not deep from the outer limbus. And these experiments, because we see cells with the same color after such a long term, strongly suggest that the inner limbus renews the cornea and it contains stem cells, while the outer limbus mostly self-renew itself and serve as a boundary, preventing the conjunctivalization of the cornea, and that both niches host stem cells. And that's how the cornea looks 4 months post-induction. We see large stripes. As I said, most of them are coming from the inner limbus, while only few are coming from the outer limbus that is busy mostly with healing. Mathematical analysis showed the limbal stem cell dynamics and strongly suggested that the data fits with a model that the outer and inner limbus contains limbal stem cells that are abundant and not rare entities as previously shown, and that limbal stem cells follow stochastic neutral drift dynamics. And actually, there was also, this was done in collaboration with the lab of Yoni Savir and Lior Starinsky, which was Strinkovsky, which was the student. And a computerized simulation that they did in parallel was also published in eLife recently. And in this model, we can simulate the formation of the stripes, and we can give different features to stem cells and to progenitor cells and see how these features affect the growth and the pattern. And then we see if this fits with the biological data, and this helps us a lot in designing the next experiments in vivo and distinguishing between the two hypotheses, the different hypotheses actually. And then the last slide that I will show you data is the characteristics of the niche. So the very drastic change in gene expression between the outer limbus versus the inner limbus strongly suggests that the niche is very different. The environment that these cells are located within is very different. And indeed, we could see that lymph vessels and blood vessels are clearly abundant and visualized in the outer limbus, and they nearly don't enter the inner limbus or corneal periphery. And moreover, very interesting was the observation that immune cells, particularly several T cell types and dendritic cells, were localized also to the outer limbus, and rarely we could see few cells in the inner limbus. And then a series of experiments that we also did was to ask what is the role of these T cells that are located in the outer limbus. So we looked at skid mice and nude mice that are lacking T cells because they have a mutation that the development of T cells is abnormal, they don't have T cells. Or we could also in parallel use immunosuppressors like steroids and also other agents like antibodies. And all these experiments supported a model that the loss at the absence of T cells, we don't see at all 2 out of the 3 most prominent outer limbal stem cell markers. So they go from 100% expression to 0. You don't see it here, you can see it in the paper. And we also see that there is a loss of quiescence. So the cells in the outer limbus that typically divide slowly, now they divide twice faster in the absence of T cells. So we think that T cells are important for the marker expression. We don't know what is the role of each genes of these markers, and also for the quiescence, for the slow division rate of outer limbal stem cells. And also in their absence of T cells, there was a delayed healing of the corneal epithelium. So taken together and to sum up, I'm just hesitant actually whether to sum up or if you want to ask first questions. I don't know what you prefer, Daniel or Juliana.
No, I think it's good to summarize and then ask.
Okay. Okay, I just find an excuse to rest a little bit from talking because I'm speaking a lot. So actually, we see, if I want to summarize what I just showed you, so we propose that there are 2 discrete limbal stem cell niches, the outer limbus that is hosting abundant limbal stem cells. They mainly regenerate themselves. They self-renew the outer limbal zone alone. Probably these cells are similar to other tissue-specific boundary cells that we have in different epithelia where there is a transition in the epithelia, for instance, between the esophagus and the stomach. Okay, there is a transition. So I suppose that these kind of cells should be in any epithelium. But these cells also have probably an important role as a reservoir of stem cells because they enter proliferation upon corneal epithelial injury. These cells are regulated by T cells. These T cells are quite abundant in the outer limbus. And then in the inner limbus, we see that there's another population of stem cells. We think that these are also abundant stem cells, and these cells are renewing the corneal epithelium constantly. Of course, this model raises many questions. And also the fact that we have the atlas of the corneal epithelial states, many states in the corneal epithelium lineage, including the progenitors, the suprabasal cells, the wing cells, and the superficial cells. We can now study them. We can look for the transcription factors and the mechanisms that regulate these cells. It could be, of course, very interesting. And there are also other questions that I don't have time, and I will probably let you ask if you want. And also, the other part of the study that I showed you was the very satisfying, actually, I would say, very surprising, unexpected plasticity of committed cells. So we find something that is quite surprising in terms of limbal stem cell deficiency, right? If you deplete the entire limbal epithelium, you don't have stem cells in the corneal epithelium at all. So in that case, the cornea can still sustain somehow. And the fact that it sustains is because these committed cells can heal the corneal epithelium, okay? And they probably sense something in the niche, we don't know exactly what, and then they start to express a lot of K15 GFP, not only in the inner limbus, also in the outer limbus. This we haven't published yet. And then these cells later on go further to demarcated zones again of outer and inner limbus. And actually, similar plasticity was seen after depletion of many stem cells of different tissues, also in the gut, in the hair follicle, specific ablation by genetic tools or by laser, etc., induced de-differentiation of the committed cells. And this tells us that maybe if we will learn these signals, maybe we'll be able to produce stem cells from any cell or in the corneal epithelial lineage. Does it mean that any cell in our body that is going to be in this limbal compartment can become limbal stem cell? I think the answer is no, because committed cells are close enough so that they are able to change a bit and to become stem cells, while other cell types in our body, neurons, for instance, if they will be here in this zone, I don't think they will become limbal stem cells. And with this, I'd like to just thank all the people that did the work, and especially to Aya Lange, which is the lab manager and the research associate, and also Wasim Nasser that did the de-differentiation study. And Aya did all the studies that I showed you together with Wasim and with Anna Altschuler, a PhD student. Wasim is an MD PhD student. And also special thank to Noam Tarazi, a medical student that helped a lot in the technical assistance and also biological and in that data interpretation, and also to the collaborators, especially Yoni Savir, who did a lot of help, and others that contributed. And with this, I will stop talking and really looking forward to your questions.
Thank you very much, Ruby. It's amazing work. I think it's very dense. What is amazing is that this new knowledge has been done in 3-4 years max, and changed completely our view of the identity of stem cells and how the cornea and I guess other tissue could renew. One of the things that is really interesting is the T cells that are in the stroma just beneath the outer compartment. And this is very interesting, but also puzzling, because you know better than me that in disease like Aniridia, one of the first events is inflammation. So inflammation is also T cells, dendritic cells. So it's an apparent contradiction of, in one way, presence of T cells is here to maintain the stem cells quiescence, able to renew the tissue and activate it in case of wound. Also, it's known that the tears of patients contain inflammatory cytokines. So how do you explain? Also, you show that in the SCID mice, where the T cells are absent, it's not like Aniridia, right?
Yes, so in SCID mice, the cornea is completely transparent.
But do you think it could be due to reduction of PAX6 in Aniridia, which is not the case of the SCID or other animal model?
Yeah, so What I think actually that T cells are important. It's clearly important once you don't have them. And we did several, 3, 4 approaches. We used not only SCID mice, also SCID NOD mice. It's another model. Also mice. And we also used the dexamethasone application to inhibit the inflammatory response. And in all cases, which is 4 different approaches, you see a very robust and dramatic change in the gene expression of the outer limbus. So I think CD63 and GPHA2, these are markers of the outer limbus in mouse. They are completely gone when you look at SCID mice. They are 0. We cannot see them. So this was quite striking.
Only the markers that change, also the quiescence.
Right. So this is the marker. In parallel to that, we did some study of proliferation, EDU incorporation assay, and we saw that the outer limbal cells are not proliferating slowly as usual. They're double faster. And this suggests that the T cells induce some cytokines that induce quiescence. Of course, that if you do injury or if there is pathology, which is very complex, and if there are other inflammatory cells, the immune system is so rich, and there are so many cell types, and in different conditions they behave differently. So of course that in the context of many, this doesn't mean that any immune cell would do the same. So it probably means that this is the role of T cells. We don't know exactly which T cells, by the way, we're now studying that, and we don't know what are the cytokines that are important, or the ligands, or if it's a cell-cell interaction, or so on. And we need to study all that in order to know also the application to and the relevance to stem cells. Now I lost the...
Any idea if in an Aniridia mouse model, you have a different presence of T cells, of joint cells? And did you look at the outer versus inner compartment in Aniridia or not yet?
No, we didn't look. So we don't study Aniridia. We don't have a model of Aniridia, but I think it's quite appealing to do research in these directions, to know more about limbal stem cell deficiency, to know more about the relevance to Aniridia. And if speaking about the Aniridia, I think that the niche is very important. So we show that loss of limbal stem cells alone doesn't lead to pathology. This is very reproducible. If you do it in a lab, I think in mouse it's very easy. Probably will work in any species, but we haven't tried. It works always. You remove the entire limbal epithelium, you don't see any problem in the mice. You see the dedifferentiation, always the stripes are coming back. Or if you don't have stripes, you do histology, you see that the tissue heals. Now, since PAX6 is expressed by the epithelial cells, we think that the stroma is affected by the epithelium, and this is why in Aniridia there is an effect, and there is this pathology that in the cornea and the stem cell deficiency like.
You know that some people claim that during development, the stroma, embryonic stroma, express PAX6.
Mm-hmm.
Do you have any idea if you confirm that, and if it could explain also the appearance of Aniridia symptoms, and also maybe some early lymphocyte cells could express PAX6 as well?
I think it well could be. It well could be that PAX6 is expressed during development and also in adulthood at levels that are very low that you cannot see it, but it still we cannot exclude that it has a very important role in the stroma. My gut feeling is that since PAX6 is so highly expressed in the epithelium, and given that there is such a well-known interactions between epithelium and stroma, my gut feeling would be that it is the epithelial PAX6 that is doing the problem in the haploid insufficiency in Aniridia. But of course, it is needed to be studied, and for that, one would need to do co-cultures of stromal and epithelial cells and study the interactions in the presence of absence of PAX6, and also perhaps to deplete PAX6 only in the stroma or only in the epithelium and see if you get these phenotypes in mice and so on and so forth. It's very interesting directions, actually. Yeah.
Juliana, I can ask other questions, or there are questions in the YouTube participants.
Well, we can ask Matteo if he's on. He can tell us if we have questions from the audience.
Sure. One of them is, in your experience with Aniridia patients, limbal stem cells transplantation has proved to be helpful to keep the cornea graft clear after a penetrating or a lamellar keratoplastic in the long term.
This is a question or
Yes.
a statement?
No. This is a question. In your experience, that there is some good results in terms of transplantation of.
Yes. Right. So actually, maybe I didn't understand the question, but yes. So the limbal stem cells were grafted or limbal tissue, and as I said, it healed the opacification of the patient. So it works very successfully, even without the ability to isolate the stem cells. In the past, people could do these experiments, and it's really working well. I think that hundreds of patients and maybe more were treated by this way, and they were the results are quite promising. I think 80% were cured for quite long term. And I think that in this term, it's important to say that it is probably the grafted epithelial cells are also influencing the niche, because we see that the niche is very important, and it's apparently, even though the niche might be affected in these patients, the grafting of the limbal epithelium alone also helped without doing anything in the stroma. So this also suggests that there is a crosstalk that coming from the epithelium to the stroma.
That's okay. Another question is, what about advanced therapies about and last news about cultivated corneas, as you said before, from stem cells from the mouth and similar?
Yeah, so one of the things in stem cell biology is to understand the genetic pathways of self-renewal of stem cells. What is the mechanism that controls the self-renewal, the ability to stay long-lived and to generate more and more cells forever? Because in vivo, in our tissues, the stem cells, they proliferate all our life. They can age maybe, but still they sustain the tissue. But once we isolate the cells and put them in culture, in most tissues, nearly I think all tissues except embryonic stem cells, that this was resolved. And maybe gut stem cells were a bit resolved also to some extent in three-dimensional culture. We don't have the ability to pass limbal stem cells and other stem cells for long-term in culture. And the reason for that is that we don't understand the interaction and the signals that the limbal stem cells get from the niche. And once we will do that, we will be able to get these signals, to capture these signals. We'll probably be able to put them in our culture dish, and those cells will be growing forever, and we will be very happy, I think.
That's okay. I don't know, Juliana, if you have other
I don't know
questions.
if Daniel wanted to ask more questions or not.
Yes, I have a lot of questions, but if you have questions, please.
No, go ahead, Daniel. Thank you.
So I have a question about the boundary between conjunctiva and the cornea. So in normal situation, conjunctiva do not invade the limbus, but in aniridia or limbal stem cell deficiency, it is a case. So do you think that this stroma compartment is responsible for this boundary, or do you believe that maybe the outer quiescent cells are physically able to prevent cells to invade? What do you think about that?
Yeah, so it's like, actually the question is, what is the reason for, if I can rephrase with your permission, what is the reason for limbal stem cell deficiency? And there are a few hypotheses that I could think of. One of them would be that the outer limbal stem cells form the boundary and they prevent the invasion of conjunctival cells into the corneal epithelium. And similarly, it could be that they crosstalk and they have a conversation with the stromal epithelial, the stromal cells underneath. and also in every region you can think about the same thing. And also you can think about prevention of the angiogenesis and the vessel that usually they don't invade the inner limbus. So maybe it is the inner limbal stroma that prevents the invasion, and maybe the PAX6 mutation is due to the mutation in stroma or epithelial compartment of the inner limbus is preventing the blood vessel formation. Another hypothesis could be that loss of limbal stem cell renewal of the cornea induces this invasion of conjunctivization or the blood vessel invasion. Because the limbal stem cells without PAX6, this is just an hypothesis, right? Cannot renew the cornea. They cannot proliferate anymore. At a certain point of the life, they stop. And then in that case, the conjunctiva are healing, and then the cornea becomes opaque. So this is another hypothesis. And of course, there is an hypothesis that the anti-angiogenic hypothesis that only the blood vessels would come in the stroma underneath the stroma of the corneal epithelium, because the epithelial cells could not secrete anti-angiogenic factors. And since the blood vessels enter, the stroma change, the stiffness of the stroma or the extracellular matrix changes, and this leads to the transdifferentiation of the corneal epithelial cells. So I think there are many hypotheses. Probably I covered only few, but now when we have the outer limbus and inner limbus, we have some tools, and maybe I would encourage people that are doing the human culture more than me, and they have more access to patients and to patient samples, much more than me, to go in these directions.
And one question that should interest everyone is, I know that other people have make also the RNA-seq analysis on human tissues. Did you find there's differences of outer and inner markers in the human part? Do you think that's relevant? All this is relevant to human?
I think the lab of Linda Laco published a very nice paper in Ocular Surface recently, published a single-cell sequencing, and they see something very similar in a way. So they see markers like GPHA2 and EFIDM3 that are markers in the mouse of the outer limbus. These are also markers of these crypts that are very unique to the human limbus, and they are absent in the mouse, actually. So they see these markers of outer limbal cells. I don't know if these cells are quiescent or not. They didn't check their proliferation as far as I remember. And I don't think they or neither us could see the outer and inner zones in the human or in other species. So it would be really interesting to see, because this is also relevant to the patients, of course, whether this is also the case of having a storage of stem cells in the outer, more protected zone that is more protected because it has maybe more melanocytes. Maybe it is more remoted zone that is protected from the UV and has the scripts. So yeah, these are questions that we need to ask in the future for sure.
Okay, very interesting. Thank you very much, Ruby. Amazing.
Thank you. Thank you so much.
Thank you. So it looks like we've covered up a lot of questions. A very interesting dialogue, a lot of new ideas for research. Thank you, Ruby. Thank you, Daniel. We appreciate you being here. So before we close this session, please let me announce that we have another webinar in our program. It will take place on the 21st of June at 6. This will be titled Managing Aniridia, Do's and Don'ts from Infancy to Adulthood. And this webinar will be presented by Dominique Bremont-Gignac and Barbara Kasman-Keller. So don't miss it. Thanks again for joining us today, and we'll see you next time. Thank you.
Thank you.
Bye-bye.
Bye.